This lecture corresponds to the second half of the lecture on the nervous regulation of the circulation. What we're going to do for this last part of the lecture is to look at some of the reflex mechanisms for the maintenance of your mean arterial pressure. We're going to play a little bit more emphasis on the baroreceptor reflexes, since we are actually going to be doing that in lab, but we'll also discuss some other reflexes. Now, the baroreceptors are a type of mechanoreceptor whose job is to respond to changes in blood pressure. Changes in blood pressure causes differences in stretch of these type of receptors. So they're actually kind of type of stretch receptors. They have what we call spray type endings, where if there is a stretch, these things kind of stretch out or expand out. If that happens, they will increase their firing rate. If they're not stretched, the nerve endings don't get spread out and the firing rate is less. They are located in the larger systemic arteries, especially in your neck and in your thorax. Specifically, good things to remember is you have them in your carotid sinus and also your aortic arch in heavy concentration. Now, this here shows you the carotid sinus. This is distinct from the carotid body. It's very important to realize that. The carotid bodies contain chemoreceptors. The carotid sinus contains baroreceptors. The aortic arch also contains a large amount of baroreceptors. The aortic body up here, like the carotid bodies, contain chemoreceptors. These baroreceptors sense changes in pressure, and they send their information up to the vasomotor center through two different pathways. The ones in the aortic arch, they move up through the vagus nerve, sending the sensory information up to the vagus nerve, up through to the nucleus tractus solitarius, up to the medulla, to the vasomotor center. The carotid sinus sends information up through Herring's nerve, then cranial nerve 9, which is your glossopharyngeal nerve, through the nucleus tractus solitarius, then eventually up into the vasomotor center. These receptors respond to pressure changes. The carotid sinus and the aortic bodies both respond to pressure ranges of 120 millimeters of mercury. But you'll notice in your notes that the carotid sinus responds to pressures between 60 and 180 millimeters of mercury, while the aortic bodies respond to pressures from 90 to 210 millimeters of mercury. The carotid sinus, a good way to remember the fact that it responds to a little bit lower pressure, is that the carotid sinus is very important in protecting the blood flow to your brain. So you want to make sure that thing's really working if your blood pressure gets too low, because if you don't get blood flow to the brain, you're going to have some issues. Both the carotid sinus and the aortic body work best around your mean arterial pressure, but they obviously will respond for a range of 120 millimeters of mercury. They respond better to rapid changes in pressure, and they work a little bit better in responses to increase in pressure than a decrease in pressure, but they're still going to work. What I want to show you is kind of what happens if your mean arterial pressure increases. So on this picture to the far left, this shows you your normal pressure range. Here's your systolic, here's your diastolic. Your mean arterial pressure, according to Guyton, runs about 100 millimeters of mercury, but it's actually less than that. Around your normal blood pressure, this is your baseline frequency or the firing rate of the baroreceptors. If the pressure increases, you'll notice that the firing rate increases. So the baroreceptors are going to fire at a greater rate. That's going to send more impulses up to the vasomotor center. If the blood pressure drops, like you see over here, you have a decreased frequency. of those action potentials coming from the baroreceptors and less impulses going to the vasomotor center. So let's go through a particular example of what could happen if your blood pressure changes, either increases or decreases. So the first example is something happened to cause your mean arterial pressure to increase acutely or rapidly. The baroreceptors are going to stretch out. In that happen, they have those spray type endings. They increase their firing rate. So in response to an increase in pressure, the firing rate from these baroreceptors increase, sends increased action potentials, frequency of action potentials, to the A2 area of the vasomotor center, which was the sensory area. It's receiving that sensory information. The firing rate from these is going to increase. And remember, the A2 area of the vasomotor center sends out axons to C1 and A1. But A2 inhibits one of these areas and activates another. So if A2 is more active, what's going to happen is it stimulates the A1 area of the vasomotor center, which was also referred to as the vasodilator area. This area innervates the heart. With an increased firing rate from A1, you're going to have an increased firing rate to the heart. And what's going to happen? You want you to think again. A1, think parasympathetics. It's going to decrease heart rate. If you decrease heart rate, what will that do to your cardiac output? Since cardiac output is equal to heart rate times stroke volume, that will decrease your cardiac output. Cardiac output is one of the components of your equation for mean arterial pressure. So if you decrease cardiac output, that's going to help to get blood pressure back down to normal. In addition, besides the increased firing rate from A1, you will have an inhibition of C1 because A2 inhibits C1. Also, remember A1 also inhibits C1. So firing from C2, which is the vasoconstrictor area, will decrease. If you have decreased firing from C1, what will that do? It, remember, innervates blood vessels. These are your sympathetics. But here, we're decreasing the firing from the sympathetics to the blood vessels. So the blood vessels, the smooth muscle is going to relax and vessels will dilate. If blood vessels dilate, resistance will decrease. If resistance decreases, that will also serve to decrease your mean arterial pressure. The C1, less firing rate from C1, you'll have decreased sympathetic activity on the heart, so heart rate will decrease. Your contractility will also decrease, which decreases your stroke volume. Both of these will result in a decreased cardiac output. A decrease in cardiac output leads to a decrease in mean arterial pressure because mean arterial pressure is equal to cardiac output times total peripheral resistance. So if you think about it, you had some change in pressure. In this case, blood pressure went up. You want to get that blood pressure back to normal. So what means can you do it? I can decrease resistance by dilating blood vessels. I can decrease my cardiac output by decreasing heart rate and decreasing cardiac contractility. And you see all of these together helping to get the mean arterial pressure back down towards normal. Well, what happens if you have a decrease in mean arterial pressure? And this will be a specific example of what we will do in lab. I'm going to have you lie down on the tabletop in lab. I'm going to ask you to get baseline heart rate, get a baseline blood pressure, and you're also going to be looking at your pulse pressure recording on your finger as a means to see what happens to blood flow to your finger. And it will be an indirect way of looking at what happens to resistance. So what will I ask you to do is to stand up as quickly as you possibly can. As soon as your feet contact the floor, I want you to record blood pressure. I want you to record heart rate and also the pulse pressure recording. Well, what's going to happen is when you first stand up, all the blood is going to rush to your feet. What will happen is your blood rushes to your feet, your venous return will decline. You'll have less blood going to your heart. So if you have a decrease in venous return, your cardiac output will decrease. If your cardiac output decreases, your blood pressure will decrease. So as soon as your feet contact the floor, your blood pressure has dropped. But the baroreceptors are very quick in picking up that drop in blood pressure. What they do is, with a drop in blood pressure, their firing rate will decrease. So you have less firing from the baroreceptors up to the A2 area of the vasomotor center. So this, the firing rate from A2, will decrease. So you have a decline in the firing rate from A2. What's that going to do to your vasodilator, A1? What will that do to the vasoconstrictor area? So I have less firing from A2. I'm going to have less activation of A1. So the firing from A1 will decrease. If you have decreased firing rate from A1 to the heart, think again, parasympathetics, heart rate, you're letting off the brake. Your heart rate is going to increase. If your heart rate increases, that will help to increase your cardiac output, and that will help to get the blood pressure back up. At the same time, you have the increased firing from C1 because A2 is not inhibiting C1 as much now. And so C1's firing goes up. That's your vasoconstrictor area. This is your sympathetics. This is going to result in constriction of your blood vessels. It's going to result in contraction of the vascular smooth muscle. If you constrict a blood vessel, your resistance will increase. If resistance increases, that will help to increase your mean arterial pressure. Also, because of its effects on the heart, increased sympathetic activity on the heart results in increased heart rate and contractility. So you have effects on heart rate, and because of the effect on contractility, stroke volume goes up. Both of these will result in an increase in cardiac output, which also again will help to increase your mean arterial pressure. So when you stood up, your blood pressure dropped, but the baroreceptors kicked in very quickly to cause changes in your cardiovascular system to get that mean arterial pressure back up towards normal. If this does not happen, or it's not as quick or significant, you'll have people that pass out if they stand up too quickly, or you have people that say, oh, they get a little bit lightheaded when they stand up too quickly. People have what we call postural hypotension, with changes in posture, and specifically standing up. Their blood pressure drops and they have a problem getting the blood pressure back up. Now in lab, when you do this, the majority of you will not detect the drop in blood pressure because this is so quick to act. What you will detect and record in lab will be the baroreceptor reflex. So you'll see the changes in heart rate. You'll see resistance increasing. And the way you're going to see that in the lab is that you're going to see the amplitude of the pulse pressure recording getting smaller, which indicates the blood vessels have constricted and you've decreased flow to that skin. You will also detect an increase in mean arterial pressure. Initially, it'll be kind of an overshoot. It's like, because it's not exactly perfect. It's like, oh, let's get that blood pressure up. You kind of a little overshoot, maybe higher than normal, but then very shortly after you're standing there, it's going to get down back towards that normal value. As you continue to stand, your blood pressure will stay slightly elevated, because usually when you stand, your blood pressure is a little bit higher than when you lie down, just because of the effects of gravity. Now these baroreceptors are definitely important when we're talking about postural changes, so from lying down to standing up, which is what we're going to be doing in lab. The baroreceptors, they say, have a buffer action. Think of when in biochemistry you learn about acid-base and balance and learn about chemical buffers like the bicarbonate buffer. Those buffers are used to resist changes in pH. Well, the baroreceptors act as buffers, but their buffer action is to resist changes in mean arterial pressure. However, if your blood pressure remains steadily increased, it's not like an acute change in blood pressure, like it's slowly increasing or slowly decreasing over time, the baroreceptors can actually reset themselves, where then they become sensitive around that new pressure that you're at. So you see this like with people that, say, for example, have high blood pressure. Over time, their blood pressure stays elevated. The baroreceptors adapt to actually work around that blood pressure. So if they do their postural changes or do things that cause their blood pressure to increase or decrease over their new higher blood pressure, the baroreceptors will work to get that blood pressure back to that new higher blood pressure. So they say that the baroreceptors are good for acute control of blood pressure, but not the long-term regulation of blood pressure. Even though more and more evidence is coming out that the baroreceptors still may play a role, but there's not enough evidence right now to really definitively say that they play a significant role in any long-term regulation of blood pressure. The kidneys are going to play a more major role in that. The chemoreceptors, which I had mentioned earlier, which are found here in the carotid body and the aortic body, they respond to changes in oxygen levels, hydrogen ions, carbon dioxide. But you can put a big X through this because the chemoreceptors have more of an effect on respiration rather than blood pressure. So when I talk about the respiratory system, that's when I'm going to talk about the chemoreceptors a little bit more in depth. So you can just ignore those for purposes for this try, or at least for this topic. Now, other type of reflex that we have is we have something called an atrial and pulmonary artery stretch reflex. In your notes, you have in bold that these are low pressure receptors that respond to stretch. Now, I need to explain what they mean when I call them low pressure receptors. They are receptors located in the atria and in the pulmonary artery, which happen to be areas of low pressure. They respond to changes in volume, which elicits different changes in stretch. They don't respond to changes in blood pressure like the baroreceptors. So they just happen to call them low pressure receptors because in these areas of the body, the pressures are relatively low. So what happens is if you have increases in volume that are in the atria or in the pulmonary artery, this elicits a reflex that's going to result in increases in urine output. So how is it going to do that, and what's the ultimate result? It works by affecting the kidneys. In the kidneys, what it will do is it causes your afferent arterioles in your nephrons to dilate. These are your feeder vessels to the glomeruli. So you'll increase the amount of blood going to the nephron. And if you dilate the afferent arteriole, what that do is it allows you to, and you can fill this in, increases your filtration. So you have an increase in filtration. If you filter more, what will happen is urine output will increase. So you're going to excrete more urine. The volume will increase. But think about it. You had an increase in volume. I want to get the volume back down. So you have an increase in urine output, which what will it do to plasma volume is it will decrease plasma volume. Now, what could happen to blood pressure if you decrease plasma volume? Well, blood pressure could drop because plasma volume is a part of your cardiac output. So blood pressure could actually drop as a result of this. This reflex involves your hypothalamus. That increase in volume in the atria and the pulmonary artery sends information to the hypothalamus, and the hypothalamus is where you produce a hormone called antidiuretic hormone. It's produced in the hypothalamus, but it's stored in the posterior pituitary. But what happens is the increase in volume says, hey, hypothalamus, don't make as much antidiuretic hormone. Don't secrete it. Well, antidiuretic hormone, what it does, think of what antidiuretic means. When you take diuretics, people take it so they increase their urine output to get, they have some fluid retention. Antidiuretics do the opposite. It causes you to not retain, antidiuretic hormone causes, I call it the anti-peeing hormone. It causes you to retain fluid. So what this is happening here is you're decreasing ADH. So I'm not going to retain as much fluid, and so you start increasing your urine output. So you're able, the nephrons going to have a lot more fluid leaving the nephrons. You're not going to be reabsorbing it back into your bloodstream. So you have an increase in urine output, and that also will lead to a decrease in plasma volume. So what's the significance or the purpose of this volume reflex is it prevents overload of the atria and the ventricles. It's like too much volume, don't want to get a little bit too much overload. So it says, hey, let's decrease the volume a little bit, and it works through the kidneys. Directly, like you see over here, or indirectly by its effects by affecting ADH release. Another reflex that is important is something called a Bainbridge reflex. It's similar to the atrial stretch that we talked about when we talked about the intrinsic regulation of cardiac output, whereby you have an increase in volume. It stretches those P cells in the atria, which causes your heart rate to increase. But with this one, this is an actual reflex. The atrial stretch thing was an innate part. It's inherent to the heart itself. Well, this one, you've got an increase in volume in the atria, results in stretch of the atria, but information is going to go up through the vagus, up to the vasomotor center, and cause information or cause an effect on your pacemaker of your heart. So motor output is going to go out. Efferent information is going to go out. And what's going to happen is it will cause will result in the inactivation of the beta-1 receptors in the SA node. And if you activate the beta-1 receptors in the SA node, what's going to happen? That will increase your heart rate. So what happens is you get an increase in heart rate, and this is even more than that innate or that intrinsic atrial stretch that we talked about earlier. So instead of like a 15% increase in heart rate you get with just stretching of those P cells, you got a 40 to 60% increase in heart rate using the Bainbridge reflex. So this is kind of a significant reflex, and again, it responds to changes in volume. Now, if I have an increase in heart rate, again, what could happen to your blood pressure? Your blood pressure can increase. But the kind of idea here with this Bainbridge reflex could be kind of very similar to the atrial and pulmonary artery stretch reflex. Is if you've got an increase in volume, and say, hey, start getting out. I don't want things backing up. Trying to move that volume out. The last topic that we're really going to go over, because one last thing you notice we're not going to go over very well or very much, but this is the next or the last big thing that we're going to talk about, and it's not going to really take much time, is the role of the central nervous system in the control of blood pressure. There's two things that are very significant in their effects on your blood pressure. There's something called the CNS ischemic response and Cushing reaction. Both of them have the same effects on your blood pressure, but the trigger is different. So the CNS ischemic response, you can kind of figure out what triggers it. Ischemia in the central nervous system. But what triggers the ischemia is if your mean arterial pressure drops below 60 millimeters of mercury. That's pretty low mean arterial pressure. So if your mean arterial pressure drops below 60 millimeters of mercury, this triggers a CNS ischemic response, because it's going to say, hey, my blood pressure is not going to be adequate enough that I'm going to get adequate amount of blood flow to my brain. So what this does is this triggers a huge mass sympathetic response. And they say a mass sympathetic response of the highest order. What will happen is you're going to restrict flow to pretty much everywhere. We're talking even the kidneys. You're going to cut off, and that's not a good thing to have to cut off blood flow to the kidneys, because those kidneys need to filter your blood. But there's like, I don't care about anybody. I need to really constrict blood vessels so I can get blood pressure up sky high, so I can make sure I can get enough blood flow to the brain. Now this is kind of like a last ditch type of effort. This is a very significant thing to happen, because you cut off blood flow, restrict significant amounts of blood flow to other parts of your body. That's not a good thing. But here's the last ditch effort to make sure you get enough blood to go into the brain. The Cushing reaction, you get the same type of response, but the trigger is if you have compression of the cerebral circulation. So maybe you have some edema, you're producing a little bit more cerebral spinal fluid, you've got an injury to the brain. So somehow you get compression of some blood vessels within the brain. That will trigger a mass sympathetic response, again very significant, to get that blood pressure up, because it's saying, hey, I'm compressing a blood vessel. I could try to really increase pressure to force fluid through it. And so that's what it's trying to do. So both the CNS ischemic response and the Cushing reaction have very significant effects on trying to get blood pressure up high enough to make sure you have adequate blood flow to the brain. The question is, why would they do this? I mean, you're shutting down blood flow to the kidneys, the liver, other places, except for, like, real core areas. Well, the reason why they do it is what would happen with prolonged ischemia in the brain. If you get prolonged ischemia in the brain, you got maybe 3 to 10 minutes before neural cells are lost forever. The neural cell, pretty much neural cells, you lose them, they're gone. You don't replace them. And so you don't want prolonged ischemia in the brain. So the central nervous system is going to be instrumental in if you've got problems with maintaining adequate amount of blood flow to the brain. There's some special means of controlled blood flow by the nervous system, which we'll kind of encounter when we talk about exercise. There's something called the abdominal compression reflex that is important, say, during exercise, is when you're exercising, you have increased muscle tone. You tend to compress your abdominal muscles. That helps to compress venous blood reservoirs, to help increase venous return and increase cardiac output. It's kind of helpful in trying to make sure we get more blood back up to the heart and keep that cardiac output up. You also have the actual contraction of your skeletal muscles. You have a muscle pump from the compression of skeletal muscles. So from the movement, that helps to ensure, again, more return of blood back up to the heart to increase venous return and increase cardiac output. You also have sensory receptors within the muscle themselves. We talked about Golgi tendon organs in the past, and your muscle spindles which respond to stretch. These are also important during exercise, and they send information. These proprioceptors send information up to the vasomotor center and influence blood pressure. Now, something that may be kind of interesting to you is when you compare isotonic versus isometric exercise. It so happens that the blood pressure responses, the cardiovascular responses that we see during isometric exercise are coming predominantly from muscle tension, So when you're maintaining an isometric contraction, those muscles are compressing down on blood vessels. You don't have much blood flow going there while you're doing isometric. You usually can't do isometrics for very long. The information is coming from that muscle tension, and what you get is a mass sympathetic response that's a little bit more exaggerated than you see with an isotonic contraction. And so when we look at exercise at a later lecture, you're going to see what happens to someone's blood pressures in response to doing an isometric contraction versus an isotonic. Now what's going to be important is when you look at an isometric contraction, is that I said the responses that you see with the cardiovascular system in the vasomotor center is coming from muscle tension itself. So if you let go, you stop the exercise, you will not see the response. So in lab, it's very important that you take a blood pressure and record the heart rate and all the other stuff you need to record while they're actually doing the exercise. Otherwise, you will not see the response. Isotonic contractions, you can do it after they've done their thing, after they've done the exercise, because you'll see the responses stay a little bit, remain after exercise has ceased, and that's what we're actually going to be doing in lab. Now, key things that I want you to try to remember from this lecture, and I will do this, I will cover this myself in face-to-face with you, is really understanding that vasomotor center, how the changes in mean arterial pressure can result in changes that get that blood pressure back towards the normal level. So you've got to use that vasomotor center. Draw yourself out a little picture. You need to remember the effects of the autonomic nervous system on the heart, the effects of the autonomic nervous system on blood vessels, which innervate the blood vessels. It's the sympathetics, which receptors are used, which innervate the heart, which receptors are used, and what their effects are. Those are probably the hardest things, but they're really important to understand that vasomotor center.